My name is Lawrence Rosenberg, and this is the Alpha Human Podcast. Before we proceed, let me remind everyone that I am not a doctor, nor am I a medical or healthcare professional. The content of this podcast and any information, advice, opinions, or statements within it are not intended and are not to be used as a substitute for professional medical advice, diagnosis, or treatment. You should not disregard or delay in obtaining medical advice for any medical condition you may have, and you should seek the assistance of a healthcare professional for any such conditions, and always inform your doctor of any changes you make or are thinking about making to your lifestyle. Okay, so in the last episode of the Alpha Human Podcast, we talked about the fact that I uncovered 4 important findings in the medical and scientific literature from my extensive research that might have a significant impact on Chromophobe Renal Cell Carcinoma, the cancer that I've been living with and dealing with for over 7 years now, including a surgery to remove a grapefruit-sized tumor on my left kidney, along with the kidney itself, by the way, and 2 smaller 2-centimeter lesions on my right kidney, which we've been monitoring for a number of years now, as well as another surgery to remove a 2-centimeter tumor in August of last year from my liver. Wonderful, isn't it? Okay, so 2 of those findings we discussed in detail during the last episode of the podcast. And, we kind of drilled down into them. I won't go back over those today. Because in this episode, we're going to talk about the 3rd finding that I uncovered, insights gleaned from Thomas Seyfried, professor of biology, genetics, and biochemistry at Boston College. And the 4th finding, a pioneering study on a novel way to kill chromophobe cancer cells, led me to my new oncologist and author of that study, who I am now seeing in combination with my original doctor. We'll discuss that crucial 4th finding in our next episode, but today I want to focus on what causes chromophobe cancer, because I am so inspired to figure this puzzle out, and to figure out any mystery, you have to investigate not just potential strategies for navigating the challenge, but it's imperative to understand the origin of the problem as well. So what exactly is chromophobe cancer? And what the hell causes it? For that matter, what is cancer? And what causes cancer? So cancer is a disease characterized by the uncontrolled growth and spread of abnormal cells in the body. Normally, cells in the body grow, divide, and die in a controlled way, but in cancer, the normal control mechanisms fail miserably, leading to the formation of tumors and the spread of cancer cells to other parts of the body. And how does cancer develop? Well, according to the National Cancer Institute, cancer is a genetic disease. That is, it's caused by changes to genes that control the way our cells function, especially how they grow and divide. However, let me read to you from a paper entitled "Paradoxical Behavior of Oncogenes Undermines the Somatic Mutation Theory." Okay, so here we go. Quote, "The currently accepted theory on the influence of DNA mutations on carcinogenesis," essentially the somatic mutation theory, we'll call it SMT for short, "is facing an increasing number of controversial results that undermine the explanatory power of mutated genes considered as causative factors. Intriguing results have demonstrated that several critical genes may act differently as oncogenes or tumor suppressors, while phenotypic reversion of cancer cell and of tissues can be achieved by modifying the microenvironment, the mutations they are carrying notwithstanding. Furthermore, a high burden of mutations has been identified in many non-cancerous tissues without any apparent pathological consequence. All things considered, a relevant body of unexplained inconsistencies with the SMT calls for an in-depth rewiring of our theoretical models. Ignoring these paradoxes is no longer sustainable." Okay, so what this paper is pointing out as a substitute to the somatic mutation hypothesis is known as the tissue organization field theory of cancer, TOFT for short. And TOFT proposes that the primary cause of cancer is the disruption of tissue organization rather than genetic mutations. Tissue organization refers to the normal interactions among cells in the parenchyma, otherwise known as functional cells, and the stroma. Stroma are the supporting cells of an organ, which are regulated by morphogenetic fields that guide tissue development and maintenance. Morphogenetic fields are patterns of information that guide the development and behavior of cells and tissues and are thought to be generated by the physical and electromagnetic properties of cells and the extracellular matrix. These fields act as a blueprint that determine how tissue should be organized and how cells should behave within that tissue. And when these interactions are disrupted by carcinogens or other factors, cells lose their normal differentiation and polarity and acquire a default state of proliferation that leads to the formation of a tumor. In contrast to the somatic mutation theory, the TOFT views cancer as a problem of tissue architecture, comparable to organogenesis. This perspective challenges the SMT's assumptions that cancer arises from a single somatic cell that accumulates multiple DNA mutations, and that cancer is a disease of cell proliferation caused by mutations in genes that control proliferation in the cell cycle. So, I went deep into the research on trying to figure out what some of those inconsistencies with the somatic mutation theory of cancer are. Notwithstanding what the National Cancer Institute says is the cause of cancer. Again, they're on that SMT, right? They're on that genetic cause of cancer. But here are some examples of the blaring inconsistencies inherent in the somatic mutation theory of cancer. 1, there are mutations without cancers or with very low incidence of cancer, such as in aging cells or in benign tumors. For example, senescent cells can accumulate mutations, but do not become malignant, while fibroadenomas can have mutations in oncogenes or tumor suppressor genes, but do not progress to cancer, breast cancer specifically. Okay, number 2, there are non-congruent mutagens and carcinogens, i.e., substances that can cause mutations but not cancer, or vice versa. For example, ionizing radiation can induce mutations but does not always cause cancer, while asbestos can cause cancer but does not induce mutations. This point about asbestos, by the way, is a good example of how epigenetics can play a role in causing cancer. An epigenetic cause of cancer, by the way, is a change in the way genes are expressed or regulated without altering the DNA sequence. Epigenetic changes can be influenced by environmental factors such as diet, stress, or exposure to toxins. Okay, number 3, there are non-parallel metabolic activation pathways for mutagenesis and carcinogenesis, i.e., some chemicals require different enzymes to produce mutations or cancer. For example, benzoapyrene requires cytochrome P450 to induce mutations, but not to cause cancer. Okay, number 4, the fourth inconsistency. There are patterns of organ-specific effects that cannot be explained by the somatic mutation theory of cancer, i.e., some carcinogens have a preference for certain tissues or organs. For example, aflatoxin B1 can cause liver cancer, but not lung cancer, while cigarette smoke can cause lung cancer, but not liver cancer. All right, here's another inconsistency. There are cancers without mutations, or with very few mutations, such as pediatric cancers or cancers in non-dividing cells. For example, retinoblastoma can occur without any detectable mutations in the RB gene, which is supposed to be a tumor suppressor gene according to the somatic mutation theory. Okay, so while the somatic mutation theory of cancer has been the prevailing hypothesis in cancer research for several decades, these blatant contradictions suggest it is not sufficient to explain the origin and progression of cancer. Yet, the SMT is the mainstay of the National Cancer Institute in stating that, quote, cancer is a genetic disease. That's what they say, right? They say cancer is a genetic disease over at the National Cancer Institute. That is, it is caused by, quote, changes to genes that control the way our cells function, especially how they grow and divide. All right, so let's look at what the National Cancer Institute, NCI for short, has to say about what can cause these types of genetic alterations that's causing cancer. So here's what they say. Genetic changes that cause cancer can happen because of 1, errors that occur as cells divide. 2, damage to DNA caused by harmful substances in the environment. Physical mutagens such as lead, arsenic, cadmium, and mercury. Exposure to various types of radiation. Chemical mutagens like benzene, beryllium, asbestos, vinyl chloride. Number 3, mutate. By the way, vinyl chloride, if I remember correctly, that was the chemical that was all over the news recently in that spill that took place over in Ohio. Vinyl chloride, nasty stuff, huh? Okay. 3, mutations that were inherited from our parents. Okay. So it's important for me to note, by the way, that there are 2 major categories of mutations. So when we talk about mutations, there's either somatic mutations, right? So there's that somatic mutation theory, or there's germline mutations. So the main difference between the 2 is that germline mutations occur in the cells that produce eggs or sperm and can be passed down from one generation to the next, right? While somatic mutations occur in non-reproductive cells and are not inherited. So germline mutations are inherited mutations. Somatic mutations are acquired during a person's life and can lead to, supposedly, sporadic cancers due to errors that occur during DNA replication or environmental factors, including tobacco use, exposure to radiation or ultraviolet light, viruses, chemical substances, and the aging process. Unlike germline mutations, somatic mutations are not inherited and are typically confined to a specific group of cells. Okay. In contrast, the germline variant is a genetic mutation that is passed down directly from a parent to their child at the time of conception, where it becomes incorporated into the genetic makeup of the developing embryo. As the embryo grows into a baby, the mutation is copied into every cell in the body, leading to its presence in all of the individual's tissues and organs throughout their lifetime. Now, according to the Oncology Nursing Society, more than 50 different hereditary cancer syndromes have been identified that can be passed from one generation to the next. Germline variants, by the way, are far less common, accounting for about 5 to 10% of cancers. In fact, the cancer that I have, chromophobe cancer, can be inherited. There's a syndrome known as Birt-Hoggs-Dubé, right? Birt-Hoggs-Dubé, BHD syndrome. And we thought perhaps that was the potential mutation that I had. And we had it checked out. We had the genetic testing done. I do not have that germline mutation, which is good because that means my children do not have that mutation. And that would lead to them having to be checked out every year to ensure that, they're healthy. Okay. Now, besides the tissue organization field of cancer, right, the TOFT, which contradicts the somatic mutation theory, there's another alternative that we have to take into account, another alternative to the somatic mutation hypothesis. And this is evidence that suggests that cancer is primarily a mitochondrial metabolic disease. Okay, so the view of cancer as a metabolic disease originated actually with the experiments of one Otto Warburg, who published his first observations on changes in the metabolism of tumors in 1923. In fact, this scientist, won the Nobel Prize for his work in this area in 1931, right? He was not, and by the way, so he was not just out there winning a Nobel Prize for his work on cancer and this idea of this mitochondrial aspect, this mitochondrial disorder or metabolic disease aspect to cancer. He was not just out there winning a Nobel Prize for that all the way back in 1931. This guy was nominated for a Nobel Prize 47 times over the course of his career. So this isn't just a fluke, right? This guy knows what he was doing. And according to Warburg's theory, respiratory insufficiency is the origin of cancer. All other phenotypes of the disease, including the somatic mutation theory, all other phenotypes of the disease, including the somatic mutations that we see, arise either directly or indirectly from insufficient respiration. And I'll explain cell respiration and why it's important in a moment, but I also want to mention that if we look back at that list of inconsistencies that I was going through, those inconsistencies with the somatic mutation theory of cancer, and we look at the point about how some cancers show epigenetic changes even in the absence of somatic mutations, what we also find, according to the paper Epigenetic Determinants of Cancer, is that epigenetic changes are present in all human cancers and are now known to cooperate with genetic alterations to drive the cancer phenotype. And according to the paper, Cancer as a Mitochondrial Metabolic Disease by Thomas Seyfried, respiration is required for the emergence and maintenance of differentiation, right? Cellular differentiation. While loss of respiration leads to glycolysis, de-differentiation, and unbridled cellular proliferation. This observation is consistent with the general hypothesis that prolonged impairment of mitochondrial energy metabolism is what underlies carcinogenesis. This hypothesis would represent an epigenetic origin of the disease in the classic sense. So with this in mind, let's now take a closer look at Chromophobe Renal Cell Carcinoma, the cancer that I have. So according to a recent paper published in the Proceedings of the National Academy of Sciences of the United States of America, quote, ChRCCs are characterized by whole chromosome losses, including chromosomes 1, 2, 6, 10, 13, 17, and 21. And get this, people, they are characterized, right, by a low mutational burden, with about 40% of cases in the Cancer Genome Atlas having no identifiable driver mutations whatsoever. Isn't that interesting? So there's a heck of a lot of chromophobe cancers out there that have no identifiable mutations. Okay. And according to a paper entitled Impairment of Gamma Glutamyl Transferase 1, Activity in the Metabolic Pathogenesis of Chromophobe Renal Cell Carcinoma, quote, Chromophobe renal cell cancers are very unusual tumors with abundant abnormal mitochondria and a low rate of driver mutations in their genomic DNA, leading to the hypothesis that the fundamental driver of chromophobe renal cell carcinoma pathogenesis involves metabolic dysregulation. Sounds like chromophobe cancer is a solid case study in support of Warburg's theory of cancer as a mitochondrial metabolic disease, and perhaps not as a genetic disorder or a genetic disease or a genetic affliction, i.e. the somatic mutation theory aspect of it. Maybe let's put aside the germline inherited theory. Let's just get back to the somatic theory of cancer. Perhaps Warburg is correct, because looks like chromophobe cancer, in many, many, many cases, doesn't have any identifiable mutations. Now, interestingly enough, turns out that in my case, I actually do have the KIT gene showing as mutated, which, by the way, is incredibly rare. In fact, I mentioned studies in the last episode that show that they never see the KIT mutation, although they see the c-KIT protein being switched on. Right? They never see the actual gene itself for that signals for this the c-KIT protein. They never actually see the gene itself turned on, which means that in all cases, by the way, where you see the c-KIT, we see the c-KIT protein being switched on, that signal just is stuck in the on position for chromophobe cancer. That lends itself to epigenetics, because for the most part, you never see the KIT gene being mutated in cancer, in chromophobe cancer. But in my case, weirdly enough, I'm one of the few, who knows, maybe the only, but certainly one of the very few that shows the KIT gene being mutated. Okay, enough of that. Then there's this, by the way, I'm going to quote again from a paper. Chromophobe renal cell carcinoma, ChRCC for short, because a lot of times I'll say ChRCC, originates from the ChRCC originates from the intercalated cells in the distal segment of the nephron. By the way, nephrons are, and we're talking about the kidney here, nephrons are the filtering units of the kidney that sifts blood and removes waste, and they play a key role in regulating extracellular fluid volume and electrolyte homeostasis. And the mitochondrial abundance present within the intercalated cells, which are the cells responsible also for regulating urine and blood pH, cause a fundamental susceptibility to mitochondrial-driven oxidative stress, creating a vicious cycle of mitochondrial damage, leading to increased reactive oxygen species, ROS, ROS for short, leading to more mitochondrial damage and thereby inducing, in many cases, ChRCC tumors, according to a paper published in the Proceedings of the National Academy of Sciences, which I'll talk more about in my next podcast. I'm not going to give you the name of the paper, because the title actually gives away the fourth crucial finding that I want to discuss, but I kind of don't want to veer off into it just yet. So I'm just going to hold off on naming the paper. So I just mentioned something called reactive oxygen species, right? So there's this, this vicious cycle of oxidative stress, mitochondrial-driven oxidative stress, which creates a vicious cycle of mitochondrial damage, which could lead to which does lead to increased reactive oxygen species, ROS, leading to more mitochondrial damage, which then lends itself to Warburg's theory. And in certain cases, that's where you could see that induction of chromophobe cancer. So what is this ROS? What is reactive oxygen species? It's a type of free radical. And we'll talk more about this free radical, ROS, as well as other free radicals in a bit. But for the moment, suffice it, because it's, by the way, we're going to talk about it because it really is at the fundamental origin of cancer. So if you haven't heard about ROS before, you're going to get to know about it now, because that's really one of the key things that's at the fundamental origins of cancer. But for the moment, suffice it to say that mitochondrial dysfunction can lead to increased levels of reactive oxygen species, ROS for short, ROS, which in turn can induce DNA damage and activate DNA repair mechanisms that can influence epigenetic markers. And while DNA damage may not always result in mutations, it can cause alterations in the way that genes are expressed through epigenetic alteration. For example, right, what I was just explaining to you about chromophobe cancer and the epigenetic change of the c-KIT protein being switched inexorably into the on position and being stuck there, inducing these changes that lead to chromophobe cancer. But here's another example, right? DNA damage can lead to changes in DNA methylation patterns, histone modifications, non-coding RNA expression, which can all affect gene expression and contribute to cancer development, even in the absence of mutations. Again, in direct contravention to the somatic mutation theory. So epigenetic changes caused by DNA damage may be transient or persistent, and can have long-lasting effects on gene expression patterns. But then again, you don't have to have DNA damage to have those epigenetic changes. Straight up, chemical toxins can cause epigenetic changes. Ionizing radiation can cause epigenetic changes. Diet, lifestyle, and as I've just went over, mitochondrial stress causing oxidative stress. Right. So we'll get into it. I'm gonna really describe this, but reactive oxygen species can cause these epigenetic changes, and a vicious cycle where more oxidative stress causes more mitochondrial dysfunction, which causes more oxidative stress and more reactive oxygen species, which causes epigenetic changes potentially leading to chromophobe cancer. Okay. Thus, mitochondrial dysfunction can contribute to epigenetic changes that promote tumorigenesis, and ergo, that's the Warburg, right? You're getting into that whole Warburg theory of cancer, which again is sounding very, very real, and is clearly a relevant theory of cancer, and it's an alternative to the genetic mutation theory of cancer. Can genetic mutations also cause cancer? Yes, but is the genetic mutation what's happening first, or is it the mitochondrial damage that's happening first that leads to DNA mutations leading to genetic damage, which leads to epigenetic changes, or even no malfunctions in the DNA code, no mutations, just epigenetic changes which cause cancer. Either way, it sounds like it very well could be it's the mitochondrial dysfunction, the mitochondrial dysregulation, which is maybe at the heart of cancer. And by the way, every cell has mitochondria, and we'll talk about what those mitochondria do. So whether cancer stems from genetic mutations, either inherited or due to environmental stressors such as chemical toxins, radiation, etc., causing tumor proliferation genes to turn on and tumor suppressor genes to turn off, or whether cancer is a metabolic disorder that begins with mitochondrial damage or adaptation, as some prefer to call it, leading to genetic alterations, the unquestionable reality is that the way mitochondria operate in cancer cells has been altered to some degree. So the question is, what is the function of normal mitochondria? So this is where we're going to dive deeper into cell respiration. I said earlier, Warburg talks about respiration and cell respiration being irreparably dysregulated. Well, what is he talking about? Well, the mitochondria, let's get into what the mitochondria are. They are organelles within the cell. Organelles are essentially mini organs, just like the body has organs like the liver, heart, and kidney, which perform specialized bodily functions. The cells themselves have tiny little structures within them that perform various jobs, like, I'll give you an example, like ribosomes are an organelle. Ribosomes are the protein factories, the protein-making factories of the cell. You have the Golgi apparatus, which prepares those proteins and also fat molecules for use in other places, in and outside of the cell. You have the nucleus of the cell, another organelle, which is the repository of genetic information and is the cell's control center responsible for DNA replication. You also have the lysosome, right? You have lysosomes, which turn out, by the way, to be crucially important in chromophobe cancer, and we'll get into that soon. Look, there's other organelles as well, but let's get back to the mitochondria, just one of those organelles. They are the powerhouses of the cell. They're responsible for producing the energy needed for cellular processes such as growth, muscle contraction, nerve impulse propagation. They do this by utilizing the energy released during the oxidation of food we eat and turning it into a fuel known as ATP, adenosine triphosphate. And the mitochondria do this via a process known as cellular respiration, of which there are 4 stages. In order, those 4 stages are glycolysis, pyruvate oxidation, the TCA cycle or the citric acid cycle, and the final stage, which is known as oxidative phosphorylation, OXPHOS for short. So during stage 1 of cellular respiration, glycolysis, glucose is broken down into pyruvate and 2 molecules of the ATP are formed. In stage 2 of respiration, cellular respiration, pyruvate is converted into acetyl-CoA. In stage 3, acetyl-CoA is turned into citric acid, which ultimately produces another 2 molecules of ATP. Now, look, I'm vastly oversimplifying the process here, but suffice it to say that between the 4 ATP produced up to this point, right, those first 3 stages, right, and that 3rd stage, by the way, is the TCA cycle, the citric acid cycle, along with NADH and FADH2, those are energy carrying molecules that during the last stage of cellular respiration, oxidative phosphorylation, transfer their energy into as many as 34 more ATP molecules. This reaction sequence, all 4 stages put together, generates a grand total of 38 molecules of ATP, the fuel that powers your cells. So every time a cell respirates, which by the way, it takes just milliseconds, 38 molecules of ATP are produced. And so every single second that a single cell respirates, it generates, get this, roughly 10 million ATP molecules. So in just 1 second, a single cell generates roughly 10 million ATP molecules. So do the math there, right? Like 38 molecules of ATP times what gets you to 10 million ATP molecules. That's how many times the cell is respirating inside a single second. And this is going on nonstop, 24/7 in every single cell in the body. To give you an idea of just how much ATP is produced daily by all your cells combined, the human body generates the equivalent of its own body weight in ATP molecules every single day. Do you know what a molecule weighs? Right? A molecule of ATP, you know what that weighs? Can you fathom just how much of that stuff the body has to generate in order for it to equal out your body weight every single day? That blows my mind. Anyway, so if the mitochondria become damaged, right, if they become dysfunctional or altered to some degree, especially, we know that in cancer cells, they are dysfunctional or they're damaged, or other people like to say they've been retrofitted or altered, or they're just adapting to the environment now in order to keep these cells alive, because cancer cells are not respirating the way they're supposed to, or even when they are respirating the way they're supposed to, what they've gone and done is they've turned some tumor suppressor genes off and tumor genesis genes on in order to help these things survive, to grow and replicate. But nevertheless, if these mitochondria are now, if they're dysregulated, if they're disordered, if their respiration is altered in some way, given that they're the damn energy generator of the cell, and the mitochondria can no longer respirate, as Otto von Warburg postulated, then how the hell are the cells surviving? Right? If they can't respirate as per normal, right? And even if they're turning certain genes on and off, even certain these epigenetic factors are now at play, creating different proteins, right? Like the key here is, they're trying to keep themselves alive, but if they can't respirate and produce fuel, then how the hell are they doing it? How the hell are they staying alive? Well, here's the answer from Otto Warburg himself. Quote, cancer, above all other diseases, has countless secondary causes, but even for cancer, there is only one prime cause. Summarized in a few words, the prime cause of cancer is the replacement of the respiration of oxygen in normal bodily cells with a fermentation of sugar. All normal body cells meet their energy needs by respiration of oxygen, whereas cancer cells meet their energy needs in great part by fermentation of sugar. All normal body cells are thus obligate aerobes, where all cancer cells are partial anaerobes. From the standpoint of the physics and chemistry of life, this difference between normal and cancer cells is so great that one can scarcely picture a greater difference. Oxygen gas, the donor of energy in plants and animals, is dethroned in the cancer cell and replaced by an energy-yielding reaction of the lowest living forms, namely a fermentation of glucose. So typically, healthy cells oxidize glucose, but what cancer cells do is they continue to survive by fermenting glucose. Whether or not there's oxygen present, it doesn't matter. They switch to this fermentation process. So how does that actually work? Like what's actually going on there when you're fermenting glucose? Well, as mentioned earlier, before entering the mitochondria for complete oxidation, glucose is first split into two molecules of pyruvate in the first stage of cellular respiration. However, if the cells cannot respirate sufficiently and as such are forced to operate in a low oxygen environment, then much of the pyruvate is reduced to lactic acid in order to maintain cell ATP production. And this is known as anaerobic fermentation, right? So, if there's a lack of oxygen, then those cells are going to switch to anaerobic fermentation of sugar to survive. But there's also aerobic fermentation. This is what Warburg was talking about, that cancer cells, they're going to switch to fermentation whether or not there's oxygen present. So instead of oxidizing glucose for fuel, right, they're still going to ferment it. And that's known as aerobic fermentation, otherwise known as aerobic glycolysis. And this involves the production of lactic acid even under normoxic conditions, right? So you have hypoxic conditions where, right, hypoxia, low oxygen, you have normoxia. So you're going to have this persistent lactic acid production in the presence of adequate oxygen. And this is indicative of abnormal respiration. Otto Warburg first proposed that all cancers arise from damage to cellular respiration. As a result, cancer cells increase their capacity to produce lactic acid even in the presence of oxygen in order to compensate for their insufficient respiration. So that's how they do it, guys. They go to lactic acid. They move to creating lactic acid, and that's that happens as a result of fermenting sugar. And that's how those damn cancer cells survive. This phenomena that cancer cells preferentially utilize glycolysis to produce energy even in the presence of oxygen is known as the Warburg effect. And this metabolic shift not only allows cancer cells to meet their energy demands, but favors, right? This metabolic shift, this fermentation, favors cell proliferation as it supports a metabolic environment and provides most of the building blocks necessary for the rapid biosynthesis that fuels growth and proliferation, i.e., those cancer cells can now continue to grow, they can survive, they can proliferate. So to summarize, according to the paper, Press Pulse, a novel therapeutic strategy for the metabolic management of cancer by Thomas Seyfried and others, the main points of Warburg's theory are: number one, insufficient respiration is the predisposing initiator of tumorigenesis and ultimately cancer. Not genetic mutations. Number two, energy through glycolysis gradually compensates for insufficient energy through respiration. And three, cancer cells continue to produce lactic acid in the presence of oxygen. And four, respiratory insufficiency eventually becomes irreversible. So now the question becomes, what causes cancer cells to switch to a permanent state of fermentation, as in the Warburg effect? So here's some reasons, right? So diving deep into the research, there's a number of reasons why your cells might switch to fermentation. By the way, this is a damn survival mechanism, believe it or not. This is so fucking crazy. When your cells become cancerous, right? When that happens, if that happens, it's actually just your cell trying to survive. Isn't that crazy? A cancer cell is a cell that survives some type of insult, typically an insult to the mitochondria. So what causes the damn insult? Number one, hypoxia. A lack of oxygen can force cells to switch to anaerobic respiration to produce ATP. Two, genetic mutations, okay? They happen, right? I mean, the SMT mutation theory of cancer is partially correct. Genetic mutations happen, and if a genetic mutation happens, it can cause cancer. The argument is over whether it's the mitochondria that goes bust first or it's the mutation that goes bust first. To me, it sounds a lot like it's the mitochondria going awry, going haywire, dysregulating first. But there are others that argue that it's the mutations that, happen first. Irrespective, I think the mutations happening first has kind of been proved in a number of cases as not being the progenitor of cancer, because then that should happen in all cancers. And the fact is, it doesn't happen in all cancers. And my cancer is one of them. Not me particularly. I'm one of the few that actually has a genetic mutation in chromophobe cancer. But chromophobe cancer is a case study in the Warburg theory because so many of them have no genetic mutations. Okay. But nevertheless, genetic mutations are an insult. Mutations in genes involved in cellular respiration can impair respiration and lead to the switch to anaerobic respiration. Number three, another insult are nutrient deficiencies. So deficiencies in nutrients, right? Deficiencies in your diet, deficiencies in nutrition required for proper cellular respiration, such as thiamine, can lead to metabolic dysfunction and the switch to anaerobic respiration. Number four, oxidative stress. Shit, here's the bitch of it. Oxidative stress is going on all day long, every second of every day when cells respirate. Those four stages, when you get down to oxphos, oxidative phosphorylation, right? Those mitochondria, when they're generating ATP, they're causing oxidative stress. They're causing reactive oxygen species that can damage the mitochondrial membrane and impair We'll talk about how that happens, by the way. How ROS can damage the mitochondrial membrane and impair respiration, leading to the switch to fermentation. Environmental toxins, right? There's another one for you. Exposure to toxins such as heavy metals and pesticides can damage mitochondrial function and lead to the switch to fermentation of sugar. Another one, viral infections. Some viruses can cause metabolic dysfunction in cells and force them to switch to fermentation. To survive, cancer is a way, it's an escape route for the cell that should die. The damn cell should just die. And in many cases, they do. That's why most of us, I shouldn't say most of us, but that's why a lot of us never get cancer. But when they don't die and they figure out how to escape death, they do it by becoming cancerous. They do it by switching to fermentation of sugar, and then that is what can lead to cancer. So viral infections can cause this as well. Chronic inflammation, right? Prolonged inflammation can lead to metabolic dysfunction and the switch to fermentation of sugar so that cells can survive. Man, I'll tell you what, that one is really scary, because you know how many people are chronically inflamed just from their diet alone, right? And that can easily lead to cancer. Finally, aging. Something none of us ever escape, no matter how good our diet is, no matter how much we protect ourselves from viruses, environmental toxins, and nutrient deficiencies. None of us can escape aging. And as cells age, mitochondrial function can decline, leading to the switch to fermentation of sugar, so that those cells, our cells are just like us, right? They are us. They're trying to survive. And in survival, they can become cancerous. How sick is that? What a joke. All right. Now, so back to Seyfried's press pulse strategy paper. I'm going to quote again, a protracted reliance on fermentation causes oxidative stress, leading to the production of reactive oxygen species, mostly through the mitochondrial coenzyme Q couple. In addition to their role in oncogenic signaling, excess ROS production damages mitochondrial function and can be both carcinogenic and mutagenic. And the somatic mutations and genomic instability seen in tumor cells thus arise from a protracted reliance on fermentation energy metabolism and a disruption of redox balance through excess oxidative stress. Right? So essentially what they're saying in this paper here, what Seyfried's saying is that all these somatic mutations that you see in cancer cells are the downstream effects of mitochondrial dysfunction, not the other way around. So we mentioned ROS a lot here, reactive oxygen species. So let's dive deep into what the hell that is. What is its role in cancer? What exactly are reactive oxygen species? So ROS are a specific type of free radical that contains oxygen, such as superoxide and peroxyl, right? So peroxyl radical, superoxide, as well as non-radical molecules such as hydrogen peroxide and singlet oxygen. These are all free radicals. When molecules split into single atoms that have unpaired electrons, they become unstable free radicals. And because electrons, they like to be in pairs, these free radicals scavenge the body. And they look to steal electrons from other molecules of proteins, of DNA, and from cellular membranes, so that they too can become a pair via a process known as oxidation. So in normal circumstances, the body provides free radical scavengers known as antioxidants, which you've all probably heard of, right? Antioxidants, right? Everyone goes around eating all these different berries to get more antioxidants. Such as, give you two examples. Alpha lipoic acid is a big one, and glutathione is another one. You'll hear more about glutathione because it's actually directly involved in a lot of cancers, including mine. But nevertheless, these free radical scavengers, known as antioxidants, they provide the needed electron and they neutralize free radicals. But if these scavengers aren't available, or if there's an overproduction of free radicals, such that they overwhelm the antioxidant supply, then these free radicals are going to donate to or steal an electron from another molecule, leading to a chain reaction that triggers the formation of more free radicals. This process is known as oxidative stress, right? Which can end up damaging proteins, altering enzyme reactions, damaging DNA, causing mutations, and lipid peroxidation. What is lipid peroxidation? This is when free radical oxidation damages the lipids, also known as fats, that form our cellular membranes. Our cellular membranes are made of fats, otherwise known as lipids. And that membrane separates and protects the interior of the cell from the outside environment. But left unchecked, these free radicals can lead to cell death and a variety of disorders, including neurodegenerative diseases, chronic inflammation, autoimmune issues, and cancer. Right? Cancer. In fact, according to a paper entitled The Interplay of Reactive Oxygen Species, Hypoxia, Inflammation, and Sirtuins in Cancer Initiation and Progression, ROS, reactive oxygen species, are at the early origin of cancer. Radiation, UV, xenobiotics, chemical carcinogens, nutrients, and chronic inflammation are sources of mitochondrial and systolic ROS. In the nucleus, they inflict damage to DNA, and in different ways they do it, including the production of random mutations, including those that allow a normal cell to lose control of the cellular cycle and of apoptosis. By the way, that's programmed cell death. So again, these cells they're supposed to die, right? They're supposed to go through this process of programmed cellular death so that they don't end up fucking you up. And that's known as apoptosis. Most of the time, mutations are correlated by one of the DNA repair mechanisms, such as double strand break, repair, double strand break repair, DSB, base excision repair, mismatch repair, and possibly nucleotide excision repair. Alternatively, the cell can undergo apoptosis, programmed cellular death. However, in these conditions, the chance of selecting a transforming combination of mutations are substantially increased by defective DNA repair mechanisms, by predisposing germline mutations, and by defective ROS detoxifying systems, i.e., free radicals. In conclusion, ROS mediate the mutagenic action of a number of carcinogenic agents, playing a prevalent role in the initial transformation of a normal cell into a tumor cell. So, we're getting right down into the origin of cancer here, guys. This is what happens. So when you hear about, this causes cancer, that causes cancer, this chemical causes cancer, radiation causes cancer, what's actually happening? This is what's happening. Oxidative stress is happening. ROS overproduction is happening. And this is damaging the cellular membrane. This is damaging the DNA. This is what's causing the cells, the mitochondria to go haywire and to cause cancer. So the overproduction of ROS and oxidative stress can either trigger the switch to fermentation, or it can be a consequence of fermentation. And although most cancer cells rely on fermentation or glycolysis to fuel themselves, we have to keep in mind that this is a very inefficient means of energy production, as glycolysis only results in the production of 2 molecules of ATP per molecule of glucose metabolized. So instead of the 38 ATP molecules healthy cells gain when they complete the oxphose stage 4 of cellular respiration, you're left with, this really inefficient mechanism for producing energy, for the cells producing energy. So how the hell do these damn cancer cells compensate if they're broken and they're, and look, I get it. They find in some cancer cells that oxidative phosphorylation is actually all well and fine. It's actually occurring. And I think they believe that oxphose is normal because they're seeing oxidative stress taking place. They're seeing the creation of reactive oxygen species. However, according to others, such as Seyfried, they're mistaking the reactive oxygen species production for oxidative phosphorylation and not the oxidative stress that's actually causing the switch to the fermentation of glucose and the mitochondria to go dysregulate. Again, chicken or egg, just put it all aside. The bottom line is this: how the hell do cancer cells compensate when they are in this state of perpetual glycolysis and fermentation? Well, cancer cells compensate for the inefficiency of glycolysis by upregulating glucose transporters on their cell surface to increase glucose uptake, even in the presence of low glucose levels. So they're able to get more out of the damn glucose. They also increase the rate of glycolysis by upregulating enzymes involved in the glycolytic pathway. So what starts to happen is these cancer cells love glucose, man. They start to suck that shit in like candy, man, like a 2-year-old eating candy. They're pulling glucose into the cell like freaking crazy. They also increase the rate of glycolysis, right? By, as I said, right, they upregulate those enzymes so they can suck in more glucose. This allows them to generate ATP and the metabolic intermediates needed for biosynthesis of macromolecules, such as nucleotides and amino acids. That's right. The metabolic intermediaries needed for biosynthesis of macromolecules, such as nucleotides and amino acids, can be created from glucose. How do you like that? That's what's required for rapid proliferation of these cancer cells. In addition, cancer cells can also use alternative metabolic pathways to generate energy and biomass. Chief among them is amino acid glutaminolysis, glutaminolysis in particular, glutaminolysis. What is that? So here's the deal, man. Glutamine is an essential nutrient for cancer cells due to its role as a key substrate for a number of metabolic pathways required for cancer cell growth and survival. Although glutamine is required as an alternative fuel for the tricarboxylic acid cycle, the TCA cycle, to produce ATP, right, the citric acid cycle, it also contributes to a wide range of pathways in cells, producing amino acids, nucleotides, and fatty acids, mTOR activation, and the hexosamine biosynthesis pathway, the HBP, as well as playing an important role in reactive oxygen species, homeostasis, which is key in my cancer, by the way, Chromophobe Renal Cell Carcinoma, and which we'll explore further in just a bit. Now, one of the primary functions of glutamine in cancer cells is to provide carbon and nitrogen for nucleotide and amino acid biosynthesis, which are essential for DNA replication and protein synthesis, respectively. Moreover, glutamine can be used to fuel the TCA cycle and generate ATP, right? So if the cancer cell isn't getting enough glucose, which, it's actually want to do, no problem. Here comes glutamine to the rescue, which can be used to fuel the TCA cycle and generate ATP, which is, right, that's what's required for cellular energy metabolism. So the increased demand for glutamine by cancer cells is due to their reliance on these metabolic pathways for survival. In fact, the increased demand for glutamine by cancer cells is so damn great that they become addicted to this amino acid, right? So just like the cell can become addicted to glucose in cancer, hey, if there's not enough of that, no problem. They're going to become super addicted to glutamine, to this amino acid, because it can do everything that glucose can do, right? It has such versatility as a substrate that it can be used to support multiple aspects of cancer cell metabolism and survival. So another crucial role of glutamine, by the way, in cancer cells is to provide a source of reducing equivalents in the form of NADPH, which can be used to counteract oxidative stress and maintain redox homeostasis. Glutamine can be converted to glutamate, which can then be used to generate NADPH through the action of enzyme glutamate dehydrogenase. And the high demand for NADPH in cancer cells is required for their rapid growth and proliferation. So you got all these pathways that glutamine can help with. Additionally, glutamine plays a crucial role in the HBP, as I said earlier, the hexosamine biosynthesis pathway, by providing the necessary precursor molecule. The HBP plays a critical role in cancer cell metabolism by providing a key substrate for the synthesis of nucleotides, glycolipids, and glycoproteins. The pathway is dependent on glutamine, which is a major source of the amino sugar precursor UDP, GIC, NAC. And I'm just giving you that because if I start getting into the full name of that thing, let's see if I can do it. Uridine, diphosphate, and acetylglucosamine. That's not so bad. I can do it. Right. So this pathway is dependent on glutamine because glutamine is the major source of the amino sugar precursor uridine, diphosphate, and acetylglucosamine, which is essential for the modification of proteins and lipids, which is absolutely necessary for proper protein folding, proper protein signaling, and cellular adhesion. Right? Those cancer cells, they need to be adhesive and stick, right? So that they can accumulate and form that damn tumor, right? And by the way, cancer cells are notoriously unadhesive. They're right, which is a problem, because if they do break away from the primary tumor, that's how they get loose in the body, in the bloodstream, in the lymph system. And when those damn things get loose, they can travel to another organ, which is known as metastasis. That's when your cancer spreads. Anyway, where are we? In cancer cells, I could talk some more about the HBP, because it's often upregulated in cancer, right? So this increases the damn production of that UDP, GIC, NACC, and the subsequent modification of those proteins and lipids, which has a profound impact on cancer cell behavior, right? by altering the cell signaling and the interactions within the extracellular environment, and regulating the activity of other metabolic pathways, such as the pentose phosphate pathway and glycolysis, which are important for cancer cell metabolism and survival. So glutamine is King freaking Kong here. And by the way, it also plays a role in cancer cell signaling and regulation because it can modulate the activity of signaling pathways such as mTOR, right? So you've got mTOR, which is a key regulator of cell growth and metabolism. And it can also regulate the expression of genes involved in cancer cell survival and proliferation. Other amino acids that have been shown to be important for cancer, besides glutamine, but nowhere near to the impact level of glutamine, are serine and glycine, right? So serine and glycine are important for cancer cell growth because they're essential building blocks for the synthesis of nucleotides, which are the building blocks of DNA and RNA. Cancer cells require a large amount of nucleotides to support their rapid proliferation, and the availability of serine and glycine can be limiting for this process, which provides methyl groups for DNA methylation and other cellular processes and is involved in the production of nucleotides and amino acids, as well as in methylation reactions. So cancer cells often have an increased demand for these one-carbon units. Methionine. There's another amino acid, and you'll hear a lot about some people go so far as to go on these methionine restricted diets, right, to starve the body of methionine. I mean, good luck, right? Methionine is important for cancer cell growth because it's required for protein synthesis, which is necessary for cell proliferation. So additionally, methionine is a key source of methyl groups for cellular methylation reactions, which are important for the regulation of gene expression and other cellular processes. Some cancer cells have been shown to be particularly dependent on methionine, and restricting methionine availability has been proposed as a potential strategy for cancer therapy in certain cancers, right? But like I said, man, good luck restricting methionine, because what you're essentially restricting is any food that contains amino acids. And when you're doing that, you're talking about restricting protein from the diet. And yeah, you could restrict protein from the diet to the degree that you're getting like super minimal, like half a gram per kilogram of body weight, or I think in these cases they look for 0.4 of a gram of protein per kilogram, 2.2 pounds of body weight. I mean, that's very little protein. You're talking about maybe 40 grams of protein for an average human, average male. Good luck, man. And does it really do anything? Does that really change the game? I don't know. Nevertheless, another amino acid that's important for cancer is arginine. Arginine is important because it's required, another one that's required for protein synthesis, right? And it also plays a role in the regulation of cellular signaling pathways. And in particular, arginine is a substrate for the production of nitric oxide, which is involved in the regulation of cell proliferation, angiogenesis, which we talked a lot about in my last episode. Cancer cells love angiogenesis because these cancer cells are typically hypoxic environments, low oxygen environments. They need to create new pathways, right? New vascular pathways to suck in blood and oxygen and to suck in nutrients. So they form all of these ancillary, vascular appendages, and they do it through a process known as angiogenesis. And there's a lot of cancer treatments which look to block angiogenesis and starve the tumor of the nutrients and kill the tumors. Anyway, back to arginine. Arginine, right? It's a key for the production of angiogenesis and immune function as well. So cancer cells have a high demand for arginine, and some tumors have been shown to express reduced levels of argininosuccinate synthase, which is an enzyme involved in arginine biosynthesis, leading to arginine auxotrophy. Okay, leucine's another big one. Leucine is important for cancer because, once again, like these other amino acids, it's key for protein synthesis. And it also plays a role in the regulation of cellular signaling pathways. In particular, leucine is a key activator of the mammalian target of rapamycins, right? That's the mTOR pathway, which is key for turning on protein synthesis, which again, a lot of people are freaked out, when, trying to avoid cancer or in the longevity circles. They're always looking to mute mTOR signaling, protein synthesis signaling, right? But, again, it's like, yeah, you can kill cancer by starving the body of everything, right? You need to starve the body of glucose. You need to starve the body of protein. In starving the body of protein and glucose, you could starve the body of glucose. There is no need for glucose exogenously, meaning you don't have to eat any sugar because the body makes all the damn sugar it ever needs. So good luck starving the body of sugar. If the body needs sugar, it'll just turn to gluconeogenesis, which we'll talk about in a moment, create all the sugar it wants from the protein that's in the body. And if you try to starve it of protein, guess what it does? It'll just break down the muscle tissue that you have, turn that into amino acids through gluconeogenesis, create all the sugar it wants, create all the amino acids it needs. So you want to starve cancer of these things, good luck. It'll just cannibalize the body. So anyway, and by the way, that's not to say that these strategies don't have an effect. Maybe it has an effect in slowing down certain cancers, but it's not going to kill the cancer. All right, leucine. All right, so that's another one that's super important. However, here's the kicker to all of this. Good luck, starve yourself of methionine, of leucine, arginine. Here's the thing. According to Seyfried, with the exception of glucose, right, and glutamine, none of these other potential fuels needed for tumor cell fermentation would likely be available in sufficient quantities anyway to drive robust tumor cell growth. And beyond that, here's the fucking punchline. All of these damn amino acids can be synthesized from glutamine through various biosynthetic pathways in the cell anyway. So, like I said, good luck trying to starve yourself of methionine. Other important alternative and interoperative pathways that help cancer cells gain nutrients for survival, growth, and proliferation, right, are as follows. I mentioned gluconeogenesis. I kind of explained what that's all about, right? Essentially, that's the body breaking down muscle tissue, turning it into amino acids, and then turning it into the substrates that it needs for cancer cell proliferation. Lipid metabolism, right? Fat metabolism. That's another pathway for cancer. How about that? Starve yourself of the damn glucose, starve yourself of the protein, but lipid metabolism, fatty acid metabolism, and the peroxisome, the main organelle within the cell used for fatty acid oxidation, is involved and is upregulated in many cancer types and can provide an important source of energy and building blocks for cancer cells through the production of acetyl-CoA, which can be used as a substrate for energy production within the damn mitochondria, right? You've heard me say acetyl-CoA before when we're talking about respiration. Well, you can get all the acetyl-CoA you need from fatty acid metabolism, from fats. So cancer cells can upregulate their fatty acid metabolism to increase the production of acetyl-CoA, which can fuel their growth and proliferation even under nutrient-poor conditions. Unbelievable, huh? Fucking cancer cells. They're like cockroaches. Lactate uptake, that's another way of the body, of the cells, the cancer cells getting nutrition outside of glucose and glutamine, which are the two major ones, right? Cancer cells can take up lactate produced by neighboring cells. So even if they can't produce the damn lactate that it needs from fermenting enough sugar, they'll just take it from other fucking cells and use it as a source of energy. I've mentioned the pentose phosphate pathway, so we don't need to get into that. And then this is a sick one. Endocytosis and autophagy, which is super important in my form of cancer. So here's the thing, man. Don't restrict yourself in a diet of glucose, restrict the glucose you're taking in, restrict the protein you're taking in. Guess what, man? These cancer cells, they figured out another way. Endocytosis is a cellular process that involves the uptake of substances from the extracellular environment by forming a vesicle around the substance and bringing it into the cell. Endocytosis can occur through various mechanisms, including receptor-mediated endocytosis, macropinocytosis, phagocytosis, pinocytosis, which is, by the way, pinocytosis is that's another type of endocytosis that involves the uptake of extracellular fluid and small particles such as amino acids, glucose, and lipids for nutrition. In contrast, phagocytosis, what I just mentioned, is a type of endocytosis that can involve the uptake of large particles such as other cells or debris. That's the kicker. It is a cannibal. Cancer can be a cannibal and just get its nutrition from other cells or cellular debris, broken shit in the body, broken organelles, broken cells. Using a specific receptor-mediated mechanism, both pinocytosis and phagocytosis can provide cancer cells with nutrients, growth factors, and other molecules that can fuel their survival and their proliferation. Matt, what was that? I said macropinocytosis. There's another process, that cancer cells use to obtain nutrients. This process involves the engulfment and internalization of extracellular fluid and small particles, such as those amino acids and lipids, through the formation of, instead of small vesicles, large vesicles called macropinosomes. These vesicles, they can be transported to lysosomes. Right? I mentioned that organelle that's in the cell, right? That little mini organ that's in the cell, the lysosome. Well, those large vesicles, right? The macropinosomes can then be transported to the lysosomes, and I'll talk about what the lysosomes do, for degradation and nutrient processing. And macropinocytosis is often upregulated in cancer cells as a way to obtain nutrients under nutrient-deprived conditions. Right? So it just goes, man, if it can't generate the energy it needs, it just goes to the extracellular fluid and sucks up the glucose from there, sucks up the damn glutamine from there. Right? So macropinocytosis, macro, large vesicle, pinocytosis, right? Also involves the formation of a large vesicle rather than a small vesicle. But then, you've got also... what was the one with the smaller vesicle? Maybe I'm confusing myself. There might be no, is it endocytosis, blah, 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 blah. Maybe not. Damn it. I thought one of these used a smaller vesicle. Nevertheless, let's ignore my idiocy there. Whether it's macropinocytosis or pinocytosis, they're creating these large vesicles, sucking up the extracellular fluid, shuttling it over to the lysosome, and then eating itself, essentially. So now let's talk about also another pathway known as autophagy. You've heard of autophagy. Autophagy has been bandied about as this great thing. You want, autophagy. You want to generate autophagy to get rid of all those dead cells, those broken cells. How do you generate autophagy? Fast, prolonged fast can go through this can cause this process of autophagy, so you can clean out your body of those crappy cells, those broken cells, and it's a great thing and it's a beautiful thing. Well, guess freaking what? In contrast to macropinocytosis and phagocytosis, autophagy is a process in which cells degrade their own intracellular components, such as damaged organelles, misfolded proteins, to generate energy and nutrients for other cells. Autophagy involves the formation of a double membrane vesicle called an autophagosome, which sequesters the material to be degraded and fuses itself with the lysosome, just like the macropinocytosis, transports the vesicle. This one fuses with the lysosome to form an autolysosome, and the material is then degraded by the lysosomal enzymes. And so now we get to what the damn lysosome will be, because it's really important in my cancer. Because what you'll find out in my cancer is not fueling itself the way many cancers do. So it relies on these processes like autophagy, right? Like the lysosome, right? So this is how these pathways affect chromophobe cancer. According to a paper, the decreased mitochondrial DNA content drives OXPHOS dysregulation in chromophobe renal cell carcinoma. Quote, as in many cancer types, glycolytic metabolism is enhanced in chromophobe cancer in company with the downregulation of OXPHOS found in this study, right? So we propose that, quote, the chromophobe cancer cells do feature the typical Warburg effect, right? Those cancer cells, they're trying to survive. They're switched to the fermentation of sugar. They're trying to get their energy now from this fermentation method in order to survive. But what you're going to find out soon enough is they're failing miserably in that respect. Here's another paper. Endocytosis-mediated replenishment of amino acids favor cancer cell proliferation and survival in chromophobe renal cell carcinoma. This is the punchline, guys. So, like I said, chromophobe cancer, it's trying, it's fermenting, but doesn't seem to be getting enough sugar and glutamine that way. So what does it do? Well, according to this study that was conducted with proteome and metabolome profiling of chromophobe tumors and adjacent kidney tissues, and what they did is they identified major metabolic alteration in chromophobe tumors, including the classical Warburg effect. But, quote, ChRCC has, right, they're showing here diminished oxidative phosphorylation capacity, like that other paper termed, right? So you've got downregulated OXPHOS stage 4. You got upregulated glycolytic proteins, right? So you got a higher abundance of glycolytic proteins. They're seeing that, right? So they're seeing the fermentation of sugar, typical phenomena, of the classical Warburg effect. But this study also found that there was stalled gluconeogenesis, right? That's one of those alternative pathways. It also found, right, that this stalled gluconeogenesis can be considered as one of the most relevant metabolic changes observed in chromophobe cancer. And then, right, like, okay, so you find this stalled gluconeogenesis. So the body is, the cancer cells, the chromophobe cancer cells is not using that alternative pathway. And what you're going to find is, and, we'll get to it, but what you're going to find is that the chromophobe cancer cells are not showing upregulated glucose consumption, upregulated glutamine production. Gluconeogenesis is not upregulated, so it's not attacking muscle tissue and looking to break down other amino acids for its nutrition. But the problem is that it's not getting its nutrition. It's not getting its nutrition from upregulated glucose and glutamine like other cancers. So the two primary metabolic pathways used by cancer cells to produce energy, glycolysis and glutaminolysis, fail to deliver the amount of fuel necessary for survival, growth, and proliferation. And that's, by the way, directly correlated in this paper, metabolic reprogramming and elevation of glutathione in chromophobe renal cell carcinoma, right? So I'm gonna get to that in a moment, but I want to just jump back for a second because I think I skipped over something here. Because what I wanted to talk about, again, I was kind of going into how chromophobe cancer fuels itself. I was talking about they see the typical Warburg effect, right, in chromophobe cancer, and that's not a surprise. In fact, we don't see a lot of genetic mutations, so we know that it's the mitochondria that's getting wrecked here, and causing the cells to switch to fermentation. And if we go back to Seyfried's press pulse theory here, the paper Press Pulse, a Novel Therapeutic Strategy for the Metabolic Management of Cancer, Seyfried writes, quote, that Dr. Mark Vincent, a professor of oncology at the University of Western Ontario, had suggested how, and by the way, I'm bringing this up because I want to make sure that I point out that this is my third finding, right? So I've come upon 4 findings which can affect chromophobe cancer. The first 2 I discussed, or certainly can affect my chromophobe cancer, which is why I've dedicated myself to researching this damn thing. But the third finding I really want to highlight is is found in this press pulse theory of cancer for the metabolic management of cancer, which is that, quote, Dr. Mark Vincent, right, has suggested that a press pulse strategy could be used to target tumor cells. We have now expanded in this paper, Seyfried says that they have now expanded this press pulse strategy to, quote, show how it can be used for the non-toxic management and possible resolution of cancer. That's a huge statement, guys. What they're saying is a calorie-restricted ketogenic diet or dietary energy reduction creates chronic metabolic stress in the body. This energy stress acts as a press disturbance, the effects of which would be greater in the tumor cell than in the normal cells due to the dependency on fermentation energy metabolism, mitogens, anabolic signaling, which when we talk about anabolic signaling, we're talking about mTOR signaling, IGF-1 signaling, elevated redox stress, right, which is ROS, and mutational load. So, right, by having this caloric restricted ketogenic diet or dietary energy reduction, you can create this metabolic stress, which now you get this disturbance on the cell, right, which would be greater in tumor cells than they would be in normal. So normal cells could survive the press. What they're suggesting here is potentially the failing cells, the failing cancer cells maybe couldn't take this energy stress, because it's so dependent on fermentation energy metabolism, so dependent on anabolic signaling that it can't go without it. Your healthier cells are stronger and could probably survive for a bit. And then he talks about drugs that target availability of glucose and glutamine would act as a pulse disturbance. So you've got the press, the calorie restriction, the ketogenic diet, depriving the cancer cells of their anabolic signaling. And then you have drugs that target the availability of glucose and glutamine, which would act as a pulse disturbance and cause acute reduction of tumor-dependent fuels. And he suggests hyperbaric oxygen therapy can also be considered another pulse disturbance in elevating reactive oxygen species to a greater degree in tumor cells than in normal cells, thus promoting cancer cell death through redox stress, which means that normal cells readily transition to ketone body metabolism for protection against ROS damage and oxidative stress. And so the goal, therefore, is to produce a therapeutic strategy that can far more effectively manage cancer than can the toxic cancer therapies currently used in most standards of care. So this option offers a possible alternative to the KIT inhibitors. I have the KIT mutation, but perhaps this offers an alternative to the KIT inhibitors, which may or may not work in my case should I need a strategy beyond surgery. And so this concept represents the third finding I've alluded to in a number of episodes. Now, as I've mentioned, I've been on some form of ketogenic diet for the last 7 years, which is the first prong of this method. However, if you take into account Seyfried's concept of also blocking glutamine, then this radically expands the efficacy of restricting the primary fuel that cancer uses to survive, to grow, and to proliferate, right? Which is glucose. And the ketogenic diet starves the cells of glucose. But interestingly enough, in the case of chromophobe cancer, not in the way Seyfried necessarily intends. And this is really my other finding, which we'll get to soon. But first, let's dig further into the pathways that chromophobe cancer use to gain nutrients. As although we see the Warburg effect in chromophobe cancer, weirdly enough, according to a paper entitled Metabolic Reprogramming and Elevation of Glutathione in Chromophobe Renal Cell Carcinoma, it turns out that the uptake of glutamine and glucose to sustain tumor growth was shown to be significantly lower in chromophobe cancer compared to clear cell renal cell carcinoma, right, and PRCC, or PRCC. So that right there is really, really intriguing, right? Because in chromophobe cancer, what you typically see is the Warburg effect, but you just don't see a lot of uptake of glucose or glutamine. So yeah, you can be on a ketogenic diet and starve the body of glucose, but those damn cells, those cancer cells, they're not showing a ton of glucose, which means they're not getting a lot of it from the Warburg effect, from fermentation, right? So this could be a consequence of low microvessel density observed in chromophobe cancer compared to ChRCC tumor, clear cell renal cell tumors. And that, my friends, is the kicker, because when my doctor originally told me, hey, those KIT mutation drugs, they're not going to work on you because we don't really see, not that my doctor said this, but I researched it and figured out that, oh, well, chromophobe cancer cells aren't typically angiogenic tumors. They're not creating a lot of angiogenesis. They're not getting their nutrition from, the added vasculature. And so KIT so the KIT gene, which kind of gives you a lot of vascularity so that the cells, can get that nutrition. The anti-KIT drugs, the KIT inhibitors, the tyrosine kinase inhibitors, they're not going to work on chromophobe cancer. And that's why they said, hey, look, keep relying on surgery, Lawrence, because these drugs that we typically prescribe for other kidney cancers don't really work in chromophobe cancers. But then weirdly enough, through my research, I figured out that even though they never see it, I actually have the freaking KIT mutation. So maybe the tyrosine, maybe the KIT inhibitors would work. But irrespective of whether they would or they wouldn't, right? I've been on a ketogenic diet, and it turns out that, hey, yeah, that would typically stress a lot of these cancer cells that suck up a lot of glucose. My cancer cells aren't sucking up a lot of glucose, right? And that's because they're not getting their nutrition the way a lot of these cancer cells, tumors get their nutrition, because they're not developing a lot of vasculature, right? And sucking in to suck in all of that. So if they're not creating vasculature to suck in blood and oxygen and nutrition, what are they creating? What are they doing? How are they getting their nutrition? Well, it's like I was saying earlier. This is where these fucking cells are cannibals, man. These chromophobe cancer cells. And so, like I said, they're cockroaches, man. They always find a way. And I was talking about autophagy before. We were talking about macropinocytosis, talking about pinocytosis, large, the large vesicle formation to suck in the extracellular fluid and get the nutrition that way, get it through, whether it's what's floating around in the extracellular fluid, in the form of sugar and fatty acids and amino acids, or whether it's just taking in broken cells and delivering it to the lysosome, right? And this is where the lysosome comes into play, right? So let's get into it. The two primary metabolic pathways used by cancer cells to produce energy, glycolysis and glutaminolysis, fail to deliver the amount of fuel necessary for survival, growth, and proliferation. And furthermore, according to the paper Metabolic Programming and Elevation of Glutathione in Chromophobe Renal Cell Carcinoma, almost all of the other pathways I mentioned earlier that are involved in energy supply chains and nutrition homeostasis in cancer cells, right? They're all fucking irrelevant in chromophobe cancer, whether it's gluconeogenesis, whether it's lipid metabolism, fatty acid metabolism, whether it's the peroxisome, right? Whether it's glutathione metabolism, whether it's amino acid metabolism. They're all downregulated. They're all suppressed in chromophobe renal cell carcinoma. Hence, according to this paper, chromophobe renal cell carcinoma, sorry, skipping over my words here. Let me take a sip of water. Hence, chromophobe cancer seems to be poorly supported by classic nutrition supply chains. And we get the same assertions from another paper. Just in case that was a fluke, I did more research, got another paper here. Glutathione metabolism in renal cell carcinoma progression and implications for therapies. Quote, proteome profiling has identified metabolic reprogramming in chromophobe cancer, including stalled gluconeogenesis, downregulated oxidative phosphorylation, downregulated fatty acid metabolism, downregulated amino acid metabolism. And they go on to say that as chromophobe cancer cells have a significantly lower microvessel density, right? Don't see a lot of angiogenesis there typically. They also have a lower glucose uptake because of that, especially compared to ChRCC, clear cell renal cell carcinoma, which accounts for most renal cell carcinomas, and PRCC. It seems that Chromophobe renal cell carcinoma cells prefer a different way to acquire nutrients to compensate for the nutrient-poor microenvironment. So what is that alternative pathway? Well, once again, let's go back to that paper, Metabolic Reprogramming and Elevation of Glutathione in Chromophobe Renal Cell Carcinoma. Quote, the way these, well, hold on, before I quote it, the way these damn chromophobe cancer cells are acquiring the fuel that they need to survive are, quote, through the lysosome and the proteasome, which are involved in the cell recycling machinery by contributing and delivering new biomass via endocytosis, which we talked about earlier, and autophagy, which were both significantly enriched. Right? Autophagy, endocytosis. So what are the lysosome and the proteasome? Right? Because remember, they're delivering the nutrition to the lysosome, which is an organelle inside the cell. Lysosomes and proteasomes can be found within every human cell and act as the machinery of the, they do act as the machinery of the two waste disposal and recycling systems used by our cells to detoxify themselves. Otherwise known as the ubiquitin proteasome system and autophagic lysosomal system. So how and why do the cells produce waste? Well, our cells use the 20 amino acids they get from the digestion of the protein we eat and reorganize those amino acids to create the approximately 25,000 different proteins that the human body needs for nearly every task of cellular life. As mentioned earlier, ribosomes are the organelles within the cells that act as the manufacturing plant for the generation of new proteins. And these factories use the instructions they get from the DNA sequence of a gene within the cell, within the nucleus, to build a chain of hundreds or even thousands of amino acids. And each link in that chain is one of 20 possible amino acids, and they assemble together in a unique combination to create that particular protein. Now, the extensive length of those particular proteins, and by the way, those proteins go on to do different things in the body. Now, right, because when the body needs something done, when the cell needs something done, the factory machinery goes into place, it creates a protein, and the protein goes off and signals the cell to do something. And when it's doing the wrong thing, you get cancer, right? Those epigenetic changes. Now, the extensive length, by the way, of those protein combinations make proteins sensitive to many factors, such as high temperature. They're sensitive to radiation. They're sensitive to chemicals. And all these factors damage proteins and alter their fragile structures, negatively affecting how they function. And when proteins are damaged, or when they finish performing their functions and are no longer needed, they're sent to the proteasome to be broken down. Right? And then guess what, man? The chromophobe cancer cells get to eat. Right? Unbelievable. The second waste disposal system of our cells is the autophagic lysosomal system. So here, lysosomes take care of the more complex garbage from both inside and outside the cell, such as the discarded mitochondria, right? So the mitochondria themselves, when they're done, when they're broken, when they're messed up, boom, into the lysosome for recycling. Cellular debris, metabolic products, or objects foreign to the cell, such as viruses and bacteria, they get sucked into the lysosome and it gets broken down into waste, down into the most simplest of compounds. What are the simplest compounds, right? The building blocks of protein, amino acids, the building blocks of lipids, right? The fatty acids, and of course, glucose. And they're then transferred back into the cytoplasm as new cell building materials, right? So material from outside the cell is taken up through endocytosis, while material from inside the cell is digested through a process known as autophagy. And so right? Inside the cell, the lysosome is sucking in all that broken crap, eating like a king, eating like a king. Although the thing is, it can eat like a king that way, but it does slow down, because cancer cells love to proliferate, so they love to suck in glucose and amino acids, and they grow, and they're aggressive cancers that do that really well. And then there are less aggressive cancers like chromophobe cancer, which is fairly less aggressive because of this process, because, right, it can only suck in so much, you can only cannibalize so much of itself because there's only so much broken shit in the system. Unless you fuck around and you start to go on extended fasts and you start to trigger lots of autophagy. That's the crazy thing about this, man. I'll get to that in a second. But look, chromophobe cancer does not need the typical nutrient supply chain that many other cancers use. Chromophobe cells, like the cells of a few other cancers, are getting fed directly from all the recycled crap generated by the lysosome and proteasome and related processes such as autophagy and endocytosis and macropinocytosis, pinocyte, which essentially converts all this cellular waste into amino acids, sugars, fatty acids, blah, blah, blah. And this is also why you got to be very careful with calls for long extended fasts as a way to fight cancer, because autophagy can be used by some cancers like mine as a major source of nutrients. And this is so fascinating that I'm going to have a podcast dedicated to that particular topic. But look, I think you need to look into your own cancer. This is why I stress the importance of doing your own research. I wouldn't know any of this if I haven't spent so many damn hours researching my cancer and cancer in general. Point is, if you've got cancer, look into the type of cancer you have, see how that cancer fuels itself, because a lot of people are out there telling you, go on an extended fast. It's great for your cancer. It'll hopefully it'll reduce the cancer. It'll reduce the tumor. It'll starve the cancer. But guess what, man? If your tumor is one of those that rely on cellular waste and debris, which is what autophagy kicks in, man, it kicks into play, the recycling of all the broken shit in your body. You ramp that stuff up, your cancer is going to be eaten, right? They're going to be eaten well. So this is why research is so freaking important. What's also interesting is that Thomas Seyfried mentions the lysosomal pathway, by the way, as a potential escape route for cancer cells to evade his glutamine blocking strategy. Right? So Seyfried talks about this in his Press Pulse, right? In the Press Pulse paper, or in his Press Pulse strategy. Well, I'll quote. What he says is, cannibalism and phagocytosis of cellular debris are well-known characteristics of macrophages and of myeloid cancer cells with macrophage properties. Shelton showed that, so Shelton's one of the authors of some of these papers that Seyfried has collaborated with Shelton on. Shelton showed that glioblastoma cells with myeloid properties could survive in matrigel. Shelton showed that glioblastoma cells with myeloid properties could survive in matrigel, which is the extracellular matrix material in the absence of added glucose and glutamine. The gradual accumulation of lactate in the media, it's where they're testing the cell, right? The media that they use to test the cell suggested that the glioblastoma cells survive through lysosomal digestion and aerobic fermentation of glycoconjugates present in the matrigel, in the media that they were using. Right, so they're growing cancer cells in the media. The media they're using maybe as glucose or some glucose type substance. And then what are the damn fucking cancer cells doing? They're surviving because they're digesting that shit through the lysosome. Glioblastoma cell death occurred immediately following the addition of chloroquine, which neutralizes lysosomal acidity and digestion. Shelton's findings are consistent with the more recent findings of Kamhorst et al. In showing that pancreatic ductal adenocarcinoma cells could obtain glutamine under nutrient-poor conditions through lysosomal digestion of extracellular proteins. It will therefore become necessary to also target lysosomal digestion under reduced glucose and glutamine conditions to effectively manage those invasive and metastatic cancers that express cannibalism and phagocytosis. So, there's a finding. So again, I do my research. So what's my finding? Interesting. And it's not exactly this. This is just part of my third finding, that if I not only stay on a ketogenic diet, but if I also, by the way, I'm on a carnivore diet, so I'm getting all of my nutrition, 99% of my nutrition. Once in a blue moon, I eat some strawberries. That's super rare, like not a lot. But most of the time I'm eating nothing other than fatty meat. And so I'm on a carnivore ketogenic diet. And so there's the press on the cell, right? And then the pulse would be to use some kind of drug potentially, which I haven't done, to block glutamine. And so you block glutamine, but so what? My cancer cells aren't sucking up a lot of glutamine. It's getting the nutrition elsewhere. It's getting it from the lysosome. And so one of my findings here would be that if I end up going this route, if I end up ever having to go this route, not only would I look to block glutamine, but what I would also do is I would maybe add chloroquine in small amounts or something else. I don't know. I have to do some more research into this, so don't take this as gospel. But it turns out that what they're saying here is chloroquine. Well, that will deal with the lysosomal digestion aspect of things. So again, don't take any of this as gospel, but this is fascinating. So if I'm blocking sugar, I'm blocking glutamine, and then there's also need to block the lysosomal digestion. Right? Okay. So aside from autophagy and the lysosomal pathway, there's something else that's interesting about how chromophobe cancer cells develop, how they survive, how they proliferate. And that is also at play in many other cancers, and consequently may also turn out to be the way to destroy these cancer cells, which is my fourth finding, right? It turns out that according to the paper, iron accumulation typifies renal cell carcinoma tumorigenesis, but abates with pathological progression, sarcomatoid differentiation, and metastasis. That's the name of the paper. And in this paper, quote, iron accumulation typifies renal cell carcinoma tumorigenesis. Now, what does that have to do with what causes chromophobe cancer and how to kill it? What the hell does iron accumulation? What does that have to do with how a chromophobe cancer cell develops and grows and proliferates? And more importantly, what does that have to do with how to kill it? Stay tuned. We're going to talk more about that one in the next episode of the Alpha Human Podcast. Sorry, guys. I've been talking a lot here. Again, I need a sip of water. Thank you for hanging with me throughout this episode. Hopefully I didn't bore most of you to death by getting into all of this jargon, but nevertheless, hope you got something out of it. And I will look forward to continuing this journey of discovery and research in the next episode of the AHP, Alpha Human Podcast. Remember, look at the logo behind me, right? Victory through insight, right? The Alpha Human Podcast, guys. Have a great one. Do your research.